So obviously, water balance is critical for cells and the tissues and the organs within organisms. So plant cells will lose water in hypertonic environments. And that's the situation here on the far right. Hypertonic, meaning elevated solute levels externally, but that also means lower water levels externally relative to the cells. And most solutes can't easily get across the plasma membrane, so what will happen in hypertonic environments is water, which is in a higher concentration inside the cell versus outside. Water will diffuse outside of the cells, causing cells to shrivel up. If they lose too much water, they can actually die. All right? And in plant cells, what will happen is the plasma membrane will pull away from the external cell wall. And again, if too much water is lost, it can kill the cells. So this movement of the plasma membrane away from the cell wall in plant cells is called plasmolysis. I'm going to show you a little video here of that. There's no audio, but notice you can see some of the chloroplasts moving around, but it looks like something weird is happening. It looks like inside the cells, the plasma membrane is pulling away. Actually, that is what's happening. It looks like a little spag inside the cell. It's getting smaller and smaller, but the cell wall, which is really tough and rigid, stays in place. And that's because water is being removed from the cells, and that's seen by the plasma membrane moving away from the cell wall. If too much water is lost, it can kill the plant cells. Okay? And again, that's because water is being lost by the plant cells in hypertonic environments or in hypertonic solutions. Okay, let's look at active transport. The active movement of molecules into and out of the cells. Active because it requires energy in the form of ATP. You might be able to remember this because A is in active transport, and there's an A in ATP. In fact, there's an A and a T, so active transport molecule that's driving active transport mechanism, the movement pumping of molecules across the membrane. So cellular energy in the form of ATP is used to move or pump substances or even molecules against their concentration gradient. So that's like the opposite of diffusion, and that requires energy. Sometimes cells want to build up a concentration of materials inside or outside, and to do that, they'll use membrane proteins that are referred to as pumps, and what they use in terms of energy to pump substances against their concentration gradient is the energy of ATP. So cells can use this to active transport, to maintain concentration gradient of substances inside and or outside of the cell. And that's actually how the sodium-potassium pump works. Or the sodium-potassium pump is an example of active transport, as we'll see here.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transferred protein is now ready to repeat the process.
So this is an example of active transport, the sodium-potassium pump, because the sodium-potassium pump, this membrane protein, requires ATP, energy, to move 3 sodiums out, here in yellow, and to move 2 potassiums in, here in red. And notice the effect of this. There's a higher concentration of sodium outside, and a higher concentration of potassium inside, and the sodium-potassium pump is driving sodium against its concentration gradient outwards, and is driving potassium against its concentration gradient inwards. So it's moving these ions against their concentration gradients, and that always takes energy. So let's look at exo and endocytosis. Word root, cyte or cyto means cell, exo means out of, endo means into. So exocytosis, movement of material outside of the cell. Endocytosis, movement of material into the cell. So these are for large molecules or large substances or even cells like bacterial cells that are way too big to deal with diffusion across the membrane or to get across a membrane protein channel. So exocytosis, moving material out of a cell by use of a membranous transport vesicle or export vesicle. So notice this is something that your cell has produced, and it's moving it towards the plasma membrane with an transport vesicle or export vesicle. The vesicle fuses with the membrane because the membrane of the vesicle is phospholipid bilayer, just the way the plasma membrane is a phospholipid bilayer. The vesicle fuses with the membrane, kind of the way soap bubbles would fuse together, and then what was inside gets ejected outside, so exocytosis. Remember when we talked about protein synthesis, protein for export, proteins get produced at the rough ER, transport vesicle moves the protein to the Golgi apparatus, protein gets modified, put into a transport or export vesicle that fuses with the plasma membrane, and that's material, that protein gets ejected via exocytosis. So we looked at this already. Little video.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
Okay. So endocytosis, it's like the opposite of exocytosis. If you were to run the video back of exocytosis, you'd get endocytosis, the incorporation of something from outside into the cell. The plasma membrane pinches in or invaginates, forming a vesicle that moves into the cell. So whatever got encapsulated there from outside gets moved into the cell, hence endocytosis. A special type of endocytosis is called phagocytosis. This is where a cell can engulf a big particle or whole cell, like a bacterial cell, within a large vacuole by extending the plasma membrane out with what are called pseudopodia, plural for pseudopod. Okay. So when we looked at this before, here's a food item that's getting moved into the cell. The plasma membrane invaginates, forming a food vacuole. The food vacuole will fuse with a lysosome with digestive enzymes, breaking down that molecule via chemical digestion, and then the smaller molecules are released inside the cell for things like cellular respiration. Okay, so a little video here on exocytosis. Notice that the plasma membrane kind of reaches out to grab this item and move it in, and it's something pretty large.
In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
All right. So here's a little video of what's called an amoeba This is the end of the video, so we'll see it from the beginning. But what happened is this single-celled amoeba extended its plasma membrane. Those things are called pseudopods. And it's actually engulfed two other small organisms. These are all protists. These are little ciliated guys. But they get engulfed, encapsulated into a food vacuole. And they then get digested by lysosomes, digestive enzymes within the lysosomes, within the amoeba. And even though those are relatively large, the fact that they got encapsulated, they realized too late that they need to escape. They can't. They will be ingested. Well, they have been ingested, but they will be digested internally inside this one cell. And that provides nutrients once they're broken down chemically for this single-celled amoeba. But it's phagocytosis, cell eating, because of the large vacuole that's formed by these extending pseudopodia. False feet, because they're not really feet, just extensions of the plasma membrane. And that is it for this chapter.